And I think that if you could visualize all of that together in one software suite, you'd be much better off. But we have an issue of when you try to merge all these things together into a single coordinate system, they don't always work exactly right, and there are lots of reasons behind that. But for the most part, using traditional techniques to scale a brain atlas to fit a patient, one of which is called AC-PC scaling, where you take the AC and the PC is 2 points in every person's brain. You can take an existing brain atlas and it has an AC and a PC, and you can stretch it such that it matches that of that patient. And if you do that and you lump these things on to each other or lay them onto there, that if we did this in this particular patient, the brain atlas would suggest the subthalamic nucleus is right here, but the microelectrode recording would say that the subthalamic nucleus is right here. I mean, these are pretty big differences. This is about a 10 millimeter span from probably this point to this point. So that's way off, right? The point is that using just very simple techniques to modify these brain atlases, put them into a common coordinate system, it doesn't look very promising. Well, the reason is that every person's brain is different, and in fact, until I started working on this, I had no appreciation for how dramatically different the geometry of different patients' brains really are. So how can you account for this stuff? Well, this is where Luis comes in, and he's an expert in optimization algorithms. And we have these brain atlases, and they're actually broken up into a bunch of little tiny triangles. And so when you have a bunch of little tiny triangles, you can define the centroid of each of those triangles, and then you can calculate the distance from each of these respective microelectrode recording points to each of these little triangles. And using some trigonometry, you can start to calculate whether or not these little recording points are either inside of this nucleus or whether they're outside of that nucleus, or if some of these that aren't supposed to be in this nucleus are in there and you want them to be out. And so if you can calculate those things, and computers can do these kinds of calculations lickety-split, then now we've got the basis for actually taking this information and making these 3D volumes match as closely as possible to these microelectrode recordings, and in combination with that, the MRI that is in the background here. So that's what Luis did, and he came up with these very elegant algorithms that very quickly cycle through all of this microelectrode recording data, permute the brain atlas in these sort of goal-directed searches, such that you go from this, the initial starting point or the initial conditions, to this, which is this optimized fit, where we've weighted the selection or the placement of the brain atlas to match as closely as possible with the microelectrode recording data. So why would you want to go through all of that? It seems like a hassle. If you got the MER data, then you can see where the subthalamic nucleus is. Well, nowadays they do multiple passes, several, 3. 3 is about the average, at least at the Cleveland Clinic. Well, maybe we don't need to do as many passes with the microelectrode. Every time you poke that sharp little needle down there, who knows what might happen. So if you can use this brain atlas, if you can constrain it based on the MRI and based on the microelectrode recording data, then you can use the brain atlas to tell you about the rest of the three-dimensional geometry of this target area. And so a little off, let me just go. So why would you want to do that? Well, within the atlas, you can define, sort of a consistent target location, a target location that is representative of good therapeutic outcomes. If you can put the electrode in this position in that brain atlas, and you fit that brain atlas to the next patient in line, then you've got a good deal of confidence that that next patient in the line is going to have good therapeutic benefit. So that's the concept or the motivation of why you would want to do this. Okay, well, so how does it work? in terms of comparing these optimized fits to the fits that human beings that are experienced in doing this would do. So what Luis calculated were these, sort of metrics of goodness of fit. So the distance from points that are supposed to be in the target nucleus but are outside of it, the number of points that are actually inside of their target nucleus, and points that are inside nuclei but aren't supposed to be inside their nuclei, right? So, so this is work where we're actually looking at the percent of correctly fitted microelectrode recordings. You use ACPC scaling, like I said, sort of this default technique that's commonly used in current surgical practice. Well, you're not really doing very well. Maybe we're fitting half of the microelectrode recording data accurately. That's certainly not very encouraging. We had, was it 4 or 5 expert users? 4 expert users, of which I was one, so I guess I, corrupted the data. But Dr. Walter was one of the other expert users, so he helped push this much further up, on the correctly fitted here. But you can see, when a human expert does this, we can get up to about 80% fit. You're never going to get it to work perfect. Nothing works perfect. But you take the optimization algorithm, and it can do just as well as the human experts. The difference is the optimization algorithm does it in seconds, whereas the human users need multiple minutes to perform these fits. And minutes mean something in the operating room. You don't want to waste time fiddling around with computer software when you've got someone's brain exposed. So, this is the take-home message of why we would be doing this, because it allows us to use information from this brain atlas to help us predict where we're going to get the best possible predicted outcome in that patient, and then hopefully in subsequent patients as well. So once you've put all of this into a common stereotactic coordinate system, the imaging data, the brain atlas, the microelectrode recording data, this then morphed target location from the brain atlas, you can then define this X, Y, and Z and ring and arc angles that are very specific to the placement of the frame on that patient's head, so that we can predict where we should be putting that electrode for the future. And then, here's how you could then visualize how that electrode would end up and where it would be in reference to all of that information. And the concept is that people could do this and visualize this in the operating room in combination with the current technique of using intraoperative fluoroscopy to visualize where the electrodes are. But that doesn't provide any information about the anatomy. So once the electrode's in there, then how can we optimize the selection of the parameter settings? And so this has sort of been one of the major pushes from the laboratory over the last 5 years or so. And so what we have concentrated on are developing theoretical calculations of the volume of stimulation. So this is the work primarily of Chris Butson, who's now an assistant professor at Medical College of Wisconsin, and Chris Max, who I've interesting stories I could tell you about Chris Max, but he doesn't work here anymore. So Chris Max and Chris Butson really worked on trying to build these computer models, and our motivation was based on this plot here. So this is a figure from one of the most popular papers on DBS programming, and from a very prominent German neurologist, Jens Volkmann. And so the concept is that once the DBS electrodes are implanted and the patient's sewn up, a few weeks later they will return back to the clinic, and now they are the neurologist's patient, and the neurologist and their clinical staff then are responsible for selecting the stimulation parameter settings for that patient. And this clinical selection of stimulation parameters is a pretty daunting task. We have a good idea of general parameter settings that are going to give us therapeutic benefit for Parkinson's disease. You know, for STN DBS, you know, somewhere around 2 volts, 60 microsecond pulse widths, 130 hertz stimulus. But what we don't know is what contact and what is the exact sort of stimulus titration that should be used. And unfortunately, there's very little in terms of visual guidance to help the clinician select these stimulation parameter settings. And so if you actually calculate this out of realistic stimulation parameter settings, they're well over 1,000 settings that you know, could realistically provide therapeutic benefit in any given patient. So how do you find the right one? That's a pretty big search space to look through. Well, the way that happens right now is you start with, so there are 4 contacts. You start with one, you sit there with the patient and you crank it up until you get some kind of measurable clinical effect. You mark that on some paper and you keep turning it up until you start getting some side effects, and you mark that, and then you go on to the next one, and then you go on to the next one, and you go on to the next one. So that's what's called a monopolar review, and that is, you know, used usually that first visit when the patient comes back after their surgery. And the idea is to find the right contact. So you're looking for the contact that had therapeutic benefit at the lowest voltage and had the largest window before you got to a side effect. The problem with that is that it's a very subjective process and really dependent on the experience of the clinical programmer in identifying either therapeutic benefits or side effects. And it's contaminated by the fact that stimulation is transient, and it takes time sometimes for some of the therapeutic effects to show themselves. It also takes some time for them to wear off. Same thing with some side effects. So we have lots of contaminating factors that corrupt this sort of parameter selection process. But in general, that process works very well at experienced centers. The issue is that DBS, as it's growing now from being, you know, something that's only done at very high quality clinical research centers, to now something that's done in, you know, general practitioner sort of environments that don't have everyday experience with this. This is a real problem for the growth of the neuromodulation industry. So how do we get over this problem? Well, my concept is to use neurostimulation models to actually prospectively predict where we should be stimulating in that given patient. So Chris Butson has a series of very nice papers where he has developed mathematical transitions from these computer models of the potential distribution generated in the tissue medium to predictions of the volume of tissue that's directly activated by the stimulus. And that's a function of all the different stimulation parameter settings that are available to the device. And so what we're looking at in this red blob is basically everything inside that red blob that is, or every myelinated axon inside that red blob would be suprathreshold for the generation of action potential. And any axons or neural elements on the outside of that blob would be subthreshold for action potential generation. And so that provides now a tool for us to combine with that patient's sort of computer model that we developed in the operating room. So the concept is that you would have, you know, now you put a tablet PC in the hands of this clinician program, the clinician programmer, and now they can visualize, okay, here's where the electrode ended up in that patient, and here's the microelectro recording data that we saw in that patient, and here's the 3D brain atlas that was fitted to that patient. And then in a study that I'm not really going to talk about today, but another work of Chris Butson, we spent time developing a representation for what we would consider the target volume of tissue that should be stimulated to give you the best therapeutic benefit, and that's this sort of black blob volume here. So if we have a target volume of tissue that we should be stimulating, and we have these computer models of the placement of the electrode and the volume of tissue that's stimulated as a function of the stimulation parameter settings, then we can start to calculate overlap between the predicted volume of stimulation and this target volume that we're trying to get to. And this is now where some of these optimization algorithms come into play again, because they're very good at calculating these overlaps between different volumes, and they can very quickly cycle through many, many different parameter settings to come up with a parameter setting that provides maximal filling, if you will, of this target volume and minimizing stimulation spread outside of that target volume. So here we're at 1 volt, turn it up, 2 volts. Now you can see we're getting more of that target volume, but we're also starting to spread some out. 3 volts, now we're filling almost all of that target volume, but we're also spilling a lot more out. And so you can imagine it cycling through, hundreds of these to come up with an initial prediction of, in this case, I think this was the, sort of theoretically optimal stimulation parameter setting. So,
can I ask you,
yeah.
In the previous slide,
yeah.
Shouldn't you move down one contact to match with the
Well, so the issue is that the STN, in my opinion, is not the actual electrical target of the stimulation. And that's what this, black blob is really getting at here, that there's a major fiber pathway coming right through this area called the lenticular fasciculus that in every computer model we've ever made of human subjects and monkeys is directly stimulated during therapeutic efficacy. And actually, Chris's most recent stuff, doing these probabilistic maps of where stimulation ends up, the greatest benefit is actually outside of the subthalamic nucleus. However, the sensory motor portion of the subthalamic nucleus is still, contained within this target volume here, which is this sort of dorsal and posterior, and it's you can't see it in this, but it's the lateral part as well. So, okay, so we can use these types of computer models to come up with, these theoretically optimal stimulation parameter settings customized to individual patients. But it's fun to build these models, but it's really just an exercise in sort of nerddom. So how do we actually take that and make something fun with it or do something useful with it? So in collaboration with Jay Alberts, we've been comparing clinical outcomes of our theoretically optimal stimulation parameter settings compared to the traditional or clinically selected DBS settings for individual patients. And so this study was set up really and run by Jay. And the people in my laboratory were completely blinded to anything about these patients. All we received was their imaging data and their intraoperative microelectrode recording data. And then we subsequently selected a stimulation parameter setting that Jay would clinically test in these patients. And then the clinical testing was set up such that the patients were randomized one day or the other to evaluate their clinical settings or their model predicted settings, doing some tasks I'll tell you about in a minute. There was something else I wanted to say about that study, but anyway, it'll come to me. So, the study was set up to evaluate some things that, the idea was to develop a quantitative evaluation of something that's more than just their motor effect or more than just their cognitive effect. So the task was a dual task study where they were performing a force manipulation with their finger and thumb here, where they're trying to track or do a force tracking task in combination with a working memory task. And the idea here is that clinically, DBS parameter settings are selected solely based on motor outcomes. But much of the recent data now is suggesting that there are real and measurable cognitive side effects that come from deep brain stimulation in the subthalamic nucleus. And so, so Jay developed this paradigm as a way to try and to look at both of those together, with the idea that as the cognitive demands become stronger and stronger, your ability to do things at the same time will be diminished. And that's true under any circumstances, but it's even more diminished during deep brain stimulation. And so the idea is to try and make things be similar to the way that they are in real life, where you're doing two things at once. You're talking and you're walking. You're doing cognitive and motor things together all the time. And so how can we sort of quantify that more real life situation? So like I said, the force tracking task is relatively simple. You hold and you try and monitor or maintain a certain level of force. But in combination, you're doing it with this in-back. So the in-back is a test where the presenter is saying H, 0 back, you say H, Q, Q, B, B. 1 back, the presenter says H, Q, you say H, B, Q, so on and so forth, right? So this sounds very easy, but it's not, and it can make you look very stupid quite easily. And then if you're also trying to do, something else at the same time, you can start to make some mistakes. So here's an example trace from the data. So the force tracking, you can do pretty well. You start the test, this is the target, you're trying to hold it within that. You have some confidence interval that, we're, sort of keeping it or allowing it to be within the target range. When you start doing the force tracking and the 2-back working memory task, you get flustered. You make a mistake. Oh man, now Q, Z, things get messed up and you have some problems, right? So this is an example of the data, and so now let's look at an individual patient. This is with their clinical settings. They have deep brain stimulation, which is directly stimulating the subthalamic nucleus, which is, like I said, one of the perceived targets. But unfortunately for this patient, it seems as if their clinical settings have started to spread into these more ventral areas of the STN, and these ventral areas of the STN are known to be related to limbic and associative areas, or these cognitive pathways that also flow through the basal ganglia. So the basal ganglia isn't only motor, it's also cognitive and limbic, or associative and limbic, which kind of make up cognitive activities. And so Jay's hypothesis was that patients who are having these cognitive declines are having them not because of the DBS per se, but because of DBS spreading into these cognitive circuits in addition to the motor circuits that we're trying to activate. And so when we compare this patient's clinical settings that were selected without any knowledge of cognitive evaluation to their sort of theoretically optimal settings, we see that we've moved the stimulation out of sort of these sort of bad areas down here and forced them to be higher up into the motor areas of STN and, as I said, this sort of fiber pathway area just dorsal to the STN. And we've also reduced their stimulus amplitudes quite a bit as well, suggesting that we could be, stimulating them a little more efficiently with the model. And so here's an example of that patient now sort of combining this data together. So this is the time within that force tracking target, doing force tracking only, off DBS, on their clinical DBS, and on their model-defined DBS parameter settings. And so if we just look at their UPDRS sort of clinical rating scale. Off DBS 56, that's pretty severe Parkinson's. On DBS, this is no medication, so just DBS alone, they're getting a very respectable improvement, and we're getting very similar improvements with the clinical DBS compared to the sort of optimal DBS parameter settings.
What would an able-bodied person, what would their tracking performance?
I don't know, Jay, maybe you could say a little bit about.
They probably have, like an undergraduate would have 28, 29 seconds.
And the professor? And the professor?
So, okay. So that would be, so that'd be like right here, I guess.
Yeah, they're pretty solid.
Okay. So yeah, so they're worse than normals. How's that? Okay, so so how do these things perform then as we make the task a little more difficult, right? So off DBS, you can see you start to degrade a little bit as you go from force tracking and 0 back, 1 back, and 2 back. You're getting some detriment. With the clinical DBS, you can see that really starts to take a toll. You're almost, when you're doing a complicated task, or these two complicated tasks simultaneously, you're basically back to not having DBS at all. But with the model parameters, we actually saw very little decrease in their ability to perform the task. So this was our first patient, and boy, that was really encouraging. So Jay then went on to perform those exact same evaluations in 10 more patients. So I think we have 11 total now. Yeah. So this is only 9 of them, but it gives you a pretty clean story. This is, now we're just looking at their population data with the motor clinical exam, off DBS, clinical DBS, and our model defined DBS. And you can see that we're at least able to capture the general motor outcomes that are achieved with the clinical optimization process, or the traditional clinical process. But this clinical process, these were patients that were at least 6 months out in programming. And maybe Dr. Walter can talk with us afterwards, or give us a little idea of the total amount of time that goes into programming these patients. But published studies have suggested that this is tens of hours of time that goes into this. This took a computer algorithm with no knowledge of anything about the patient. So our idea was never actually to even be able to achieve this. This was a big surprise to us. Our goal was just to get it into the right ballpark, and then the clinician would finish the job, and that's still the goal. But we were very encouraged to see that we could get as good agreement as we could, as we did with the blinded predictions. And so now we're looking at the population data with the dual task, and now we can see force tracking in 0 back, percent letters correct, 1 back, 2 back. You can see in the total population, we're seeing the same things that we saw in that individual patient. When the task is very difficult, off DBS and clinical DBS are really not performing well, but with the optimized or computer generated parameters, we're not spreading stimulation into those cognitive circuits, and we're improving their ability to do these sort of more complicated tasks.
What's the variability on this?
Actually, Jay, do you have any info on that? This is brand new data, hot off the presses. So
I don't know the exact standard deviations off the top of my head, but I know that trend was present for all patients. That's why I think we already have statistics.
So it's significant, the green one?
Yeah, the green at the FT 2 back, that was statistically significant.
So, in conclusion, I think surgical navigation, while it works very well right now, definitely has some room for improvement. And I think if we can integrate better some of these data that are already collected, we could provide a much more effective platform for visualizing DBS surgeries. I think that optimization algorithms really have a huge opportunity to speed up and improve clinical outcomes in these patients, and we have some preliminary clinical results that suggest that there is some some merit to this philosophy. So I want to acknowledge all the people in the lab that that made these contributions. Specifically Chris and Luis and Svetlana and Angela, who I highlighted their work today, and Jay Alberts and Jerry Vitek and the clinical staff that has really made this work much more than just a science project, and then the funding sources that have allowed it to happen. So thank you.
All right, Cameron, very good. Before we go to questions, we'd just like to present a small token of
thank you.
appreciation. And actually, I'll kick off the questioning. So you mentioned about your reliance on MER data collected in the operating room and some of the work that Luis is doing, and saying, can we reduce the number of tracks potentially? So have you looked at tried to look at each individual track?
Yeah.
And where's the most informative track come? Because when they're in the OR, they sort of learn from the first track and the second track. So
definitely. So so that is Luis's current project. Actually, is using the optimization algorithm to predict what would be the next most constraining track that you could do, right? And so instead of it being, it's a different philosophy than the way those tracks would be collected right now, because their goals now are to map the borders of the STN, right? So they want to define where is the lateral extent, where's the posterior extent, where's the medial extent of the STN. That's a different philosophy in every different mapper, but with the clinicians that we work with, that's their goal. But if we, made a transition to relying on this type of technology, you may think about, a different philosophy. Whereas you get an initial track, and then the next track isn't necessarily to find a border; it's to find a track that's going to constrain this brain atlas as much as possible, right? Because that's the issue. With one track, the brain atlas could still have many, many different permutations that could fit with both the MRI of that patient and those microelectrode recordings. But if you then get a second track, it becomes, there are only, so many positions now that could happen, and so on and so forth when more tracks are collected. And so, so that's what, Luis is doing now. We've also looked at, retrospectively, which track or how many tracks were necessary to get to the point where the brain atlas didn't really move around anymore. Where was the limit, so to speak, in terms of constraining it? And that was in that transition from about 3 to 4 tracks, right? Yeah. So after, once you've got 3 tracks, that's about as much information as you're going to get in terms of constraining the system. So, yeah.
Cameron, one of the things I learned to you today was almost stated as a hypothesis that, and that is that the track that is, I'm sorry, I don't know the anatomy of that certainly that well, that is, sort of between your STN and the other parts in the region of your red blob here, might be where you're really talking about targeting.
Right.
So I'm wondering if you could, so it was almost stated as a hypothesis, right? What I heard you say is in our clinical experience, this region provides the, the most clinical efficacy. So is the, would an approach, if that hypothesis is true, be to try to locate the region of that track? And I'm wondering if that's so, how would you alter the kind of mapping that you do in order to find, that specific region?
Yeah. So I think that's a very good question. In terms of how would the mapping change, I don't think it really will change. I think it will stay like this. But you're now not using this as your, sort of target. You're using this as a proxy to your target.
Gotcha.
And I think that, that is, in line with the concept of why these kind of brain atlas technologies are useful, because they provide a little more, sort of constraint, if you will, to defining those proxy targets. They have a consistent relationship between, where the subthalamic nucleus is with this sort of, zona incerta as a name that's used, or lenticular fasciculus area. They have a known relationship with each other, at least in, these generic brain data or brain atlases that are available out there. So you would use the information that you can get easily on a relative scale, STN neural recordings, but you're really using it to help constrain where the relative position is here. Does that make sense?
Yes.
Yeah.
So in musculoskeletal modeling, people have tried to do this kind of scaling for a long time.
Yes.
Sort of morphing things to imaging results, and it doesn't work very well.
Yeah.
There's qualitative differences between people, and it's just not sort of a, let's squeeze it and fit in. So have you done any, can you do really any validation of, you have this sort of functional outcome, but it doesn't really tell you that you're in the right spot, it just tells you that it worked.
Definitely.
Sort of an open loop process.
It is. This is the fundamental problem in brain analysis. There is no gold standard, aside from, when the brain atlases were originally created, where the most elegant brain atlases are created from, a patient who was imaged, in their deceased state, but, full images, then their brain's excised, then their, anatomy is sliced up, then the 3D atlas is created from that, right? Those are the most advanced brain atlases. It is verified for that one patient, but the problem is that validation there's no other way to validate it onto someone else, unless that patient then subsequently died as well, and you were able to repeat that process. How can we do it? Luckily, there are nuclei in the brain that can be seen very well under MR. So those are usually the nuclei that you would use to do this quantification process of how accurately your brain atlas did fit. But you're not getting that information for the structures that we care about. We're getting that information for other structures, and then once again you have this secondary inference. And so, the brain atlas people that work in this field as their expertise, they think that these technologies give you plus or minus 1 millimeter. That's the accepted sort of tolerance. So yeah.
I guess my question is kind of related. You start with basically channel models, right?
Mm-hmm.
You have neuron models, and you build all the way up to these FM models, and you use the first models to kind of inform all the way along. What is the effect of small errors in the beginning of this process of developing, the overlap and all this stuff?
Yeah. I think that's
and as a follow-up, I guess, what's the, if you is there one step that's really crucial in this to ensure accuracy, or is the variability sort of continuous across those?
Yeah, that's a very good question. And I don't have, a very good answer. But what I can tell you is that luckily this system is, okay, so back to this sort of channel thing and how you build up from there. It is true that, the neuron models are, gross simplifications even of the neurons themselves. And you can't always predict, with exacting threshold, the threshold for an individual fiber. But when we're talking about stimulation spreads on the order of 2 to 3 to 4 millimeters, now we're thinking on a different scale. And so while the models may have fringe effects right near threshold that are inaccurate, that's actually a very small proportion of this total red volume, right? So everything in the core of that volume is so super threshold that little inconsistencies in the models are meaningless, really. You know, it's active and it's way active, baby. You know, the ones that are questionable are right at the periphery here. But, you know, that periphery, you know, actually I would estimate that that stimulation spread error is actually less than the atlas placement error. You know what I mean? So
that was the question, right? So like how do all these errors?
Yeah, how do they compound?
And
and
it works, right? At the end, like you showed,
yeah.
Okay, so it works.
Yeah.
But
so we don't know at this point. You know, I think that the reason the subthalamic nucleus has become such a popular target for deep brain stimulation is that you can stimulate in lots of different areas around here and get nearly identical clinical outcomes. So I think the bar is pretty low. You know, we don't have to have it be, you know, like exactingly perfect. It just has to be in the right ballpark and you're going to get, you know, good effect. Now maybe in the future as these things evolve and they become more detailed, you know, we may be able to take people from getting, you know, 30 to 40% improvements in their motor scores to 50 to 60%. Maybe if we're titrating it just right, you know, we'll be able to get those benefits. But right now, you know, we don't know. And maybe that's a factor of not being able to make accurate predictions, or maybe it's just, you know, the limit of the system's already been reached.
But
So
I thought you made a pretty compelling argument that, you know, that those measures are motor, you know, and as you're looking more, recognizing more of the secondary, you know, effects of the, you know, cognitive function, you know, the bar, you are raising the bar.
Yeah, yeah.
Right?
Oh, I
know,
think so. Definitely, definitely. And, you know, and that's what will, you know, continue to happen. People, you know, are going to continue to find other side effects, you know, that were subtle at first. But now, you know, once we've learned, okay, we know how to program these devices, or, you know, the clinical community feels confident in using them. Okay, well now let's find what are the detriments and can we eliminate them? So, yeah.
You mentioned that the lenticular fasciculus might be a truer target than the SDN. Can you say some more about it? Where does that target come from? Where is it
yeah.
go? And how does that affect possible mechanisms?
Yes. So maybe if I go back to okay, so the lenticular fasciculus is a fiber tract that runs from the globus pallidus internal segment, you know, above the STN and then into the thalamus. So it is actually, you know, there are two fiber pathways that are coming from the lenticular fasciculus, or coming from the globus pallidus internus into thalamus. One is called the lenticular fasciculus, the other is called the ansa lenticularis. The ansa lenticularis is going in a different trajectory. It's not really part of this equation. But half of the output fibers from the GPI are going to thalamus right by this STN DBS electrode. And so we also know that if you put deep brain stimulators in the internal segment of the globus pallidus, you get almost identical therapeutic benefit to electrodes in the subthalamic nucleus. So the idea is that maybe STN DBS is just a different form of GPI DBS, and you know, it's more in a fiber track, which, as you know, pretty much everyone in this room knows, is easier to stimulate than, you know, sort of a nuclear bundle. So so I think that, you know, those two things start to add up, and to me that's a very compelling argument that actually, you know, we're really regulating the palatal output of the basal ganglia, as opposed to modulating, you know, the subthalamic nucleus as the principal sort of basis for what's going on at a mechanistic level. And so what does that matter? Well, the basal ganglia has, you know, sort of bursty or synchronous activity that many people think is related to the Parkinson's disease. And if you override those pathological outputs from the basal ganglia as they feed themselves into the thalamocortical system, that you then release the thalamocortical system from being corrupted by this basal ganglia input. And I think that there's a lot of support for that hypothesis. So, yeah.
So far, your results seem to be the effects of using a single contact. Have you explored the possibility of using multiple contacts in different locations, for example?
So, no, we haven't. Well, not in this study, and at least not from a clinical application. We wanted to start with, the most simple. And most patients are programmed clinically with just stimulating at one contact as well. So the idea was, let's start simple, and we can move towards more complicated or elegant stimulation strategies in the future if it works out. One of the problems with using multi-contact stimulation with the Medtronic device is that it just has a single stimulation channel. So if you use multiple contacts, and they're voltage-controlled devices, so you can only have one set stimulus amplitude, and it's voltage-controlled. So if the impedance, the electrode tissue impedance, is different at the two contacts, you're going to get different current flow out of those two contacts, but you don't know what it is because you don't know, how the impedance is changing of those contacts. So it's something that, from an engineering perspective, that I wouldn't advise. But as the technology is, sort of advancing, those things will change. And so there are new devices that will be coming out that will have multiple channels. They'll be current controlled. And now we're looking at, well, we could start to do some, current steering across, different contacts, and looking at how we could, shape those red blobs to be something more than just, spheres, if you will. So.
Cameron, how much is the electrophysiological data that is recorded from the probes able to be automated? When I've heard these, these signatures are very, very clear, but I've heard these in very, very constrained situations.
Yes, yes.
He says, here's the signature from this, here's the signature from that. You think,
yeah.
Wow, that's obvious, right?
Yeah, yeah.
But what in real life, are you able to, can you conceptually think about automating the capture of the electrophysiological data as well?
People are working on that, yes. It has, to my knowledge and what I've seen, it has not been very successful. And the reason is that, the noise characteristics are so variable. You can't anticipate, whether you're, maybe there's, more, 60 hertz noise one day than another. Maybe the background activity in that particular patient's brain is different. And so from a signal processing perspective, relying on a computer algorithm to be able to sort those things out is very difficult. The human brain is very well adapted to filter everything that's irrelevant except for the pertinent information, and it's very hard to make an algorithm do that as effectively as these neurophysiologists. So, like I said, people are working on it. I'm sure it is a nut that will be cracked, but it's not, easy. Yeah.
Right, Cameron, thank you very much.
Thanks.